Welcome to Bio 205 Microbiology at Pima Community College. In this video demo, I'm going to show you how we would perform the hand washing experiment if you were to take this in person with us. So for this particular experiment, hand washing, we're going to work here in this example with a blood agar plate, or a BAP plate for short. Your instructor could choose different types of media. They could pick mannitol salt agar if you wanted to look for staph on your skin. Other enriched media like brain heart infusion agar. So just take note in your particular lab manual of what you're working with in your class. I'm going to divide my plate down the center. And on one half, I'm going to write before. And on the other half, I'm going to write after. And so this is going to represent hand washing before. So before I hand wash, and then after I hand wash. And then you guys are going to look at some examples of this, and you're going to count the colonies before and after. And we'll talk about this in lab later. So what else do we need to put on here? Like normal, we would need our initials, the dates, the type of agar, and then in this case, we want to indicate our method. You would have a lot of flexibility in this lab. You could choose different types of chemical products. So for example, we have antibacterial soap. This is what I'm going to use. Hand sanitizers, lots of variety of these. Hibiclins, we're going to learn about this when we cover chemical methods of control, or you've already heard about it. And then this is just regular soap, so no antimicrobial properties. That would be one example. You could experiment with other things such as time, whether or not you use paper towels, etc. So in this experiment, I'm going to pick one variable, and my variable here is going to be soap, antibacterial strength. So I'm going to write that on my plate. It's not a terrible idea. If you wanted to, you could look at the active ingredients, and you could write down what was the active ingredient as well. So before we wash our hands, it depends on how many supplies you have in your classroom. You may be given, just so you guys know, one whole plate that's before and one whole plate that's after. I'm just cutting down on supplies here and using one. But before I wash, I want to touch the Petri plate and just see what's growing on my skin. So I'm going to try and stay where that black line is. I don't know if you can see it from this angle, but remember I divided it. And I'm going to rub my fingers just all over the before side. My fingers, are you can get in there. And that should be plenty. To wash our hands, we're going to turn on our water source. I like to wet my hands first. Take a pump or two of your soap or your product. Start to scrub the soap in really well. You want suds. And this is a mixture of mechanical friction, soaps, and surfactants that are going to help dislodge microbes through the action of mechanical friction. We're going to dislodge microbes from the surface of our skin here. And you generally want to do this about 20 seconds, maybe 30. And make sure you're getting in between your fingers. These are easy spots to miss. Under your nails, if your nails are dirty. Really sudsy. You could start to go up your wrist a little bit if you wanted to. It's hard because I have a lab coat on here. And then when you're finished, you're going to rinse down the drain like so. paper toweling has shown to be really an effective part of hand washing. It further removes microbes from the skin and is quite useful. So I want to make sure my hands are as dry as possible. These are not soiled with, bacterial culture, so we're just going to throw these in the trash. Using those same fingers, I'm going to go back to my Petri plate on the after side. I want to try not to dip back into the before side. And I'm going to rub my fingers again. And we're going to incubate 24 to 48 hours. And we're going to look at examples of this in D2L, and we're going to count colonies before and after and see how effective that method of hand washing was.